Good afternoon. Let's talk about the brain that rewires itself. The brain is plastic, it is changeable, and it changes with learning and experience. And that experience could be quite traumatic. Um, we're going to talk about plasticity after brain damage. Now, almost all of those who survive brain damage show some behavioral recovery. And that recovery is going to depend on the growth of new branches of axons and dendrites. And the more we can understand this process of recovery, it'll allow us to develop new and improved therapies. As somebody who has multiple sclerosis, which damages the myelin coating axons, it's very close to me to see that researchers are looking at advancing therapies for neuronal damage. Now, there's some degree of behavioral recovery for most survivors of brain damage. And this happens due to the using of different skills, but it also has to do with the rewiring of the brain or the ability for neurons to sprout new axons and grow new um, and grow dendritic spines. Some of these mechanisms of recovery are similar to those that we talked about in brain development. So again, the branching of axons and dendrites. Now, brain damage and short-term recovery. Some possible causes of brain damage: tumors, infections, exposure to toxic substances or radiation, degenerative diseases like MS or Parkinson's or Alzheimer's, and closed head injuries, which is the most common among young adults. Closed head injury refers to a sharp blow to the head that does not puncture the brain. And again, it's one of the main causes of brain injury in young adults. And the damage could range from minor to severe. Um, After severe injury, the recovery can be slow and it could be incomplete, right? So clearly, the severity of injury is correlated to length of time and regaining of function. Now, that's the most common form of brain damage for young adults, but in elderly, a stroke or cerebrovascular accident is the most common. So, a stroke is the temporary loss of blood flow to the brain, or in the case of a hemorrhage, the brain gets flooded with blood. So you can look at this figure in closer detail when you're looking at the slides, but look at the swelling of the right hemisphere in relationship to the left hemisphere. This individual died immediately after the stroke, whereas this individual lived and developed some functioning. And this, just for comparison, is what the brain of a gunshot victim looks like. There are different types of strokes. I just mentioned the hemorrhage. The hemorrhage is a less frequent type of stroke that results from a ruptured artery. So neurons are flooded with an excess of blood and therefore calcium and oxygen and other chemicals. Ischemia, which is more common, this results from a blood clot or obstruction of an artery. Neurons lose their oxygen and their glucose supply. Hemorrhages most often result in death. whereas ischemia, one is more likely to survive, and there are drugs that can be given to help with the effects of the ischemic stroke. So these types of strokes can cause an accumulation of fluid in the brain, resulting in increased pressure on the brain and increasing the probability of further strokes. It can also disrupt the sodium-potassium pump, leading to the accumulation of potassium ions inside the neuron. This excess in potassium is going to trigger the release of glutamate, which is going to lead to the overstimulation of neurons. Sodium and other ions are going to enter the neuron in excessive amounts. That's going to damage the cell, and as we know, the cell bodies of neurons do not regenerate. Excessive positive ions in the neuron block metabolism in the mitochondria and can also kill the neuron. So there is a drug called tissue plasminogen activator that breaks up the blood clots and can reduce the effects of the ischemic stroke. There's research that's begun to attempt to save neurons from death by blocking the release of glutamate, so therefore not overstimulating the cell, and reduce the entry of calcium, which again is responsible for the process of exocytosis, or the release of the glutamate. So also treatment, cool the brain. Cooling the brain reduces overstimulation and inflammation, and therefore apoptosis or cell death. Cannabinoids have also been shown to potentially minimize cell loss after brain stroke, and it's possibly due to the anti-inflammatory effects of cannabinoids. Now, the research has shown that they are most effective in laboratory animals when taken before the stroke. Okay, so like I said before, following brain damage, surviving brain areas can increase or reorganize their activity. So at first, there's decreased activity of surviving neurons after damage to other neurons. They are not receiving the stimulation that they were normally receiving. Right? One activity in one area of the brain is going to stimulate other areas. So damage to the brain disrupts patterns of normal stimulation. Therefore, we may be able to use stimulant drugs, those that release dopamine, to stimulate activity in healthy regions of the brain after stroke. So those areas that are no longer receiving incoming information can be stimulated. Now, again, destroyed cell bodies cannot be replaced. So neurons don't regenerate or regrow. But in the peripheral nervous system, damaged axons do grow back under certain conditions. So in the peripheral nervous system, if a nerve is crushed, the axon can follow the myelin to reach its previous target. And this happens at a rate of about 1 millimeter per day. However, in the mammalian central nervous system, damaged axons do not readily regenerate in the brain or the spinal cord. Scar tissue, the astrocytes, the glial cells, are going to create scar tissue, which creates a mechanical barrier to axon growth. So again, multiple sclerosis means many scars. And so the damage is to the myelin, scar tissue is created, and it blocks the ability for an axon to follow the myelinated path to its target. Now, astrocytes, why would they create scar tissue? Not entirely sure, but as they're creating scar tissue, they also produce chemicals that reduce inflammation. And you can't stop one without stopping the other. So there's research being done on how maybe to build a protein bridge so that the axon can bridge over the scar tissue. Okay. So axons, healthy axons can sprout new or branches to their axon. So an axon can grow a branch of an axon. It's called collateral sprouting when new branches are formed by the non-damaged axons, and they actually attach to vacant synapses, right? So synapse is no longer receiving a connection with an axon. There's been damage to the presynaptic neurons, and so there's a vacant space there, vacant synapse there. So healthy neighboring neurons can grow sprouts to their axons, and it can grow and take over that vacant area. Right? So cells that have lost their source of innervation release neurotrophins that induce axons to form collateral sprouts. So that what was the postsynaptic receptor that's no longer receiving information and has a vacant synapse produces this chemical that will induce neighboring axons to form collateral sprouts. And this could be great in restoring function if those neighboring neurons serve the same function as the ones that died. Then there's behavioral recovery, same behavioral recovery. If the nearby axons that innervate or grow into the vacant site played a role in some other behavior, right, that's going to be the new behavior that network is playing a role in. So over several months, sprouts fill in most vacated synapses and can be useful, neutral, or harmful. So I just told you 2 ways where it could be useful, or a way that it can be useful or harmful. Here's a figure of the collateral sprouting. Here, as you can see, there's axon from one neuron. Here's an axon for another neuron. There's damage to axon 2. It leaves a vacant receptor on this cell's dendritic branch. And here we see axon 1 grows a collateral sprout and fills in or makes synaptic connection with that empty space. Okay, so collateral sprouting can happen, and denervation supersensitivity can happen. So the postsynaptic cell, the one that's receiving the input, can develop an increased sensitivity to a neurotransmitter to compensate for the decreased amount of input that the postsynaptic cell is getting. So denervation supersensitivity is this heightened sensitivity to a neurotransmitter. Now, this can cause consequences such as chronic pain. Right? The cell becomes more sensitive to the incoming information, including pain information. I included a TED Talk on your Moodle page that I'd like you to watch. Ramachandran does a great job about describing a case of phantom limb. So a phantom limb is when somebody has a continued sensation of an amputated body part. And this happens because the cortex is reorganizing itself after the amputation of the body part, and it becomes responsive to other body parts, right? So let's say somebody has their hand amputated. The axons that once sent information to sense the hand is now there's receptor sites that are vacant. And so the axons will move into areas that have been, that are reserved for processing other types of information. And so the cortical area that is responsible for processing information from your upper extremities is close to the area of your brain to process base stimuli. And so when these axons innervate these other areas, you're still going to experience the sensation of the arm when somebody touches their face, for instance. So touch on the face can bring about the experience of the phantom arm. Use of an artificial limb can reduce the likelihood of experiencing this phantom limb. And if you watch the video, you'll see that the use of a mirror box seems to work in reducing this experience as well. So again, the phantom limb can lead to the feeling of sensations in the amputated part of the body when other parts of the body are stimulated. And here's just an example of the somatosensory cortex, the area that processes the face, the area that processes the body, and then we have amputated fingers and their axons moving into an area that processes base information. So again, if we touch this area of the face, we're going to experience phantom fingers. And again, that TED Talk does a very nice job of detailing this happening. There can also be learned adjustments in behavior when there is damage to the sensory pathway of the sensory neuron. It's called the deaffernated limb, right? So sensory neurons are afferent neurons. There's damage, so it's the limb has become deaffernated, right? The motor neurons are still working properly. However, the animal who has the deaffernated limb does not use that limb, even though they can. But there is a way to teach that animal to use that limb. So even though they can't feel that limb, they can still operate the limb, but they don't. There are therapy techniques to teach individuals to attend to that limb and to use the limb that they are capable of using. Our last slide here just shows the section of the spinal column, the dorsal roots, which are the sensory neurons, the afferent neurons that are damaged. The ventral root are the motor neurons. They remain undamaged. Again, the animal is not going to use the limb. However, if you damage the afferent or sensory pathway on one of the other limbs, they will automatically use both of them. Again, motor pathway is intact so they can move their limbs. It's a matter of what is easier and what they're not knowing what they're capable of doing. Okay. So again, the brain is capable of reorganizing itself and somewhat recovering from brain damage.